The overall goal of this procedure is to engineer freestanding nanofibers, nanostructures, and 2D matrices from single or multiple extracellular matrix proteins using surface-initiated assembly. This is accomplished by first preparing polydimethylsiloxane or PDMS stamps by pouring the PDMS prepolymer onto a topographically patterned master mold and allowing it to cure. The second step is to cut out the cured PDMS stamps and coat them with a solution containing the desired extracellular matrix proteins. Next, the stamps are washed, dried, and microcontact printed onto a poly-N isopropyl acrylamide or PIPAM coated glass cover slip. The final step is to hydrate the cover slip with warm 40 degrees Celsius deionized water and allow it to gradually cool. Reduction of the solution temperature below about 32 degrees Celsius triggers the dissolution of the thermally sensitive PIPAM layer and the release of assembled protein nanofibers or nanostructures. Ultimately, the assembled nanofibers and nanostructures are freestanding in solution as confirmed by phase contrast and/or fluorescence microscopy and can be used in further applications such as tissue engineering scaffolds.
The main advantage of this technique over existing methods such as phase separation or electrospinning is the ability to engineer ECM